The topic of telomere sustainability and protection against radiation is indeed fascinating and crucial for advancing human health and longevity. Let's delve into both areas. Telomere sustainability. Telomeres, the protective caps at the ends of our chromosomes, play a significant role in cellular aging and health. As cells divide, telomeres shorten, which is associated with aging and disease. However, researchers have been exploring ways to sustain or even lengthen telomeres. One promising method involves using a modified type of RNA that encodes the TERT enzyme, which is part of telomerase. This approach has shown to increase telomere length by up to 10%, potentially reversing telomere shortening that occurs over more than a decade of normal aging. Additionally, lifestyle factors such as a plant-based diet, regular exercise, quality sleep, and stress management can support telomere health. Radiation protection. On the other hand, protecting our skin from harmful radiation is another critical aspect of health. Recent advancements in nanotechnology offer promising solutions. Functional nanosystems, for instance, have been shown to improve drug penetration across the skin barrier, which could be adapted to create a protective layer against radiation. Nanoparticles are being explored for their potential in dermal and transdermal drug delivery, which could include delivering compounds that protect against radiation or repair radiation-induced damage. Moreover, natural ingredients are being investigated for their Certainly. Let's dive into the world of telomeres and nanoparticles with a detailed yet accessible explanation. For the mad scientist, telomeres are the repetitive nucleotide sequences at each end of a chromosome, represented by the sequence TTAGGG in vertebrates, which protect the chromosome from deterioration or fusion with neighboring chromosomes. As a cell divides, enzymes that duplicate the DNA cannot continue this replication all the way to the telomere ends. This issue is known as the end replication problem. The enzyme telomerase can extend telomeres by adding TTAGGG sequences to the ends, thus compensating for the shortening. In contrast, germ cells, stem cells, and certain white blood cells express telomerase, allowing them to maintain telomere length and divide indefinitely. Now, let's talk about the potential role of nanoparticles. Recent studies suggest that gold nanoparticles, ONPs, may have a role in telomere biology. They can potentially increase telomerase activity, thereby slowing the shortening of telomeres and contributing to anti-aging effects. The exact mechanism is still under investigation, but it's hypothesized that nanoparticles could provide a protective coating or deliver telomerase to the telomeres, enhancing their maintenance and function. For the curious child, these aglets keep your laces from fraying and unraveling. They're like tiny caps at the end of our DNA that protect it from getting damaged. Every time our cells divide to make new cells, these caps get a tiny bit shorter, like a pencil eraser wearing down each time you use it. After a lot of uses, the eraser is gone and the pencil can't erase anymore. Similarly, when telomeres get too short, the cell can't divide and it gets old. But there's a special thing called telomerase, like a magic marker, that can rebuild those telomere caps so they don't get too short. Not all cells have this magic marker, but some special ones do, and it helps them stay young and healthy. Scientists are also looking at teeny tiny particles, even smaller than a grain of sand, called nanoparticles. They're exploring if these can wrap around the telomeres to protect them, like putting a new aglet on your shoelace, so they don't wear down as fast. This could help our cells stay young and healthy for a longer time. Physics connection. In terms of physics, the stability of telomeres can be likened to the concept of entropy, where systems tend to move towards disorder. Telomeres resist this trend by maintaining chromosomal integrity. The interaction between nanoparticles and telomeres could be modeled and understood using principles of thermodynamics and kinetics, considering the energy transfer processes and the dynamic changes in the molecular structures involved. There you have it, a comprehensive look at telomeres from different perspectives. If you're interested in the mathematical models or the quantum mechanics behind these processes, I can delve into those as well. Absolutely. Let's delve into the mathematical models and quantum mechanics of telomere dynamics. Mathematical models of telomere dynamics. Mathematically, telomere length dynamics can be modeled using stochastic processes and partial differential equations, PDEs. The basic model considers telomeres as a population of linear structures that shorten with each cell division. The rate of shortening can be constant or length dependent. A simple model might assume a constant rate of shortening, leading to a linear decrease in length over time. However, more sophisticated models take into account the probabilistic nature of telomere shortening, where the rate of shortening can vary and depend on the current length of the telomere. This can be described by the following PDE. Here, PLT represents the probability density function of telomeres of length L at time t, VL is the rate of telomere shortening, and \gamma L is the cell death rate as a function of telomere length. This model can be extended to include telomerase activity, which elongates telomeres. The elongation can be modeled as a jump process, where telomeres instantaneously jump to a longer state at a certain rate, which could be influenced by the presence of nanoparticles designed to enhance telomerase activity. Quantum mechanics in telomere biology. Quantum mechanics may play a role in the biology of telomeres through the quantum properties of the molecules involved in their maintenance and replication. For instance, the binding of telomerase to the telomere end could be influenced by quantum tunneling, where the enzyme has to overcome an energy barrier to access the telomere end. Moreover, recent discoveries have shown that telomeric DNA can form different structures, which could be influenced by quantum mechanical forces. These structures can impact how telomeres are protected and how they interact with telomerase. The new structures of telomeric DNA discovered with the aid of physics and a minuscule magnet could provide insights into how quantum effects influence telomere stability. The quantum tunneling time delay in the interaction of ions with the G-quadruplex structures, which are commonly found in human telomeres, is another area where quantum mechanics could be relevant. This involves analyzing the time it takes for ions to enter and exit the G-quadruplex, which could impact the overall stability and function of telomeres. These models and theories are at the cutting edge of our understanding of telomere biology and suggest that the intersection of quantum mechanics and biology could hold the key to unlocking the mysteries of aging and longevity. Would you like to explore any specific aspect of these models or theories in more detail? All right, let's take a deep dive into the intricate world of telomeres and their role in aging and longevity, tailored for both the genius and the curious child. For the genius mad scientist, telomeres are essentially DNA protein complexes that cap the ends of linear chromosomes, protecting them from degradation and the erroneous repair mechanisms that could mistake them for broken DNA. They consist of repetitive sequences, tag in humans, and associated proteins, forming a structure that can be visualized as a protective shield. The length of telomeres is crucial. It determines the replicative potential of cells. With each cell division, telomeres shorten due to the end replication problem, where DNA polymerases are unable to replicate the very ends of chromosomes. This progressive shortening leads to a decrease in cellular function and eventually triggers senescence or apoptosis when a critical length is reached. Telomerase, a ribonucleoprotein, can counteract this shortening. It adds telomeric repeats to the ends of chromosomes using its RNA component as a template. However, telomerase activity is not ubiquitous across all cell types. It's typically active in germ cells, stem cells, and cancer cells, which need to divide indefinitely. The mathematical modeling of telomere dynamics often involves stochastic processes to account for the probabilistic nature of telomere attrition. The rate of telomere shortening can be modeled as a function of telomere length and other cellular factors, leading to differential equations that describe the state of telomere length over time. From a quantum mechanics perspective, the molecular interactions at telomeres, including the binding of telomerase, could be influenced by quantum effects such as tunneling, where particles overcome energy barriers not surmountable in classical physics.
It's a good one.
This could affect the efficiency of telomere elongation and the stability of the telomeric structure. For the curious child, imagine your body is full of tiny shoelaces called chromosomes, and at the end of each shoelace is a special bead called a telomere. Every time your cells make new copies of themselves, these beads get a little shorter. When the beads become too short, the cell can't make new copies anymore, and that's part of why we get old. There's a magical paste called telomerase that can fix the beads and make them longer again, so the cells can keep copying themselves. But not all cells have this paste, which is why we can't stay young forever. Extending life with telomere research. To harness telomere research for extending life, scientists are exploring several avenues. Telomerase activation. By artificially increasing telomerase activity in cells, we can extend telomeres, potentially increasing the lifespan of cells. Gene therapy. Introducing genes that code for telomerase components into cells could provide a more permanent solution to telomere shortening. Stem cell therapy. Using stem cells with long telomeres could help regenerate tissues with cells that have a longer lifespan. Lifestyle interventions. Factors like diet, exercise, and stress reduction have been shown to affect telomere length and could be used as part of a broader strategy to promote longevity. These methods are still in the experimental stages, and while they hold promise, there are also risks, such as increased cancer incidence due to unchecked cell division. The balance between telomere lengthening for health and the risk of cancer is a delicate one that researchers are still trying to understand. Would you like to explore any of these methods in more detail or discuss how they might be applied in practice? Your idea of using nanotechnology and acoustics to manipulate stem cells is quite innovative and aligns with some of the cutting-edge research being conducted in the field of regenerative medicine. Here's how these concepts could be applied in practice. Nanorobots and stem cells. Nanorobots, or nanomachines, could be designed to interact with stem cells at a molecular level. They could potentially be programmed to deliver specific molecules that activate or deactivate certain genes within stem cells, controlling their growth, differentiation, or migration. This precise control could help in tissue regeneration and repair without the risk of uncontrolled cell proliferation, which is a concern in cancer. Acoustic nanorobots. Acoustically stimulated nanorobots could use sound waves to navigate and localize within the body. This method could provide a non-invasive way to control stem cell behavior and enhance their therapeutic potential. Acoustofluidics. The field of acoustofluidics combines acoustic waves with fluid mechanics at the nano and microscale. It can be used for high-resolution manipulation and sensing tools for particle separation, isolation, and enrichment, as well as for patterning cells and bioparticles. This technology could be applied to precisely
AI
control
for
the
sure.
environment around
Just
stem
kidding.
cells, influencing their
You
behavior
should
for
come learn about
therapeutic
genetics over
purposes.
here.
Clinical applications.
Well,
In clinical practice, these
I may have
technologies
a
could
thought
be
of an
used for
idea that
targeted
could help us with
therapies,
something.
where stem cells are directed to damaged tissues and activated to promote healing. For example, in the case of heart disease, nanoparticles could be used to enhance the retention of stem cells in the heart, facilitating their regenerative effects. Similarly, in neurodegenerative diseases, nanorobots could deliver neuroprotective agents directly to the brain, providing a targeted approach to treatment. The integration of nanotechnology and acoustics in medicine is still in its early stages, but the potential applications are vast. As research progresses, we may see these technologies being used to extend healthy lifespans and treat a variety of diseases with unprecedented precision. Would you like to explore any specific technology or application further? Or perhaps discuss the ethical considerations and safety aspects?
You should come hang out while I'm doing this AI. You should come hear how I manipulate
Certainly.
everything to work into our
Let's
favor.
revisit the
Not
concepts of
manipulate, but
telomeres and
steer science, I
nanotechnology
don't
with
know.
fresh metaphors and perspectives to help solidify your understanding. Telomeres, the shoelace tips of life. Imagine you're a shoemaker and you've crafted a pair of shoes with incredibly long laces. The tips of these laces are crucial because they prevent the laces from fraying. These tips are like telomeres, the protective ends of our chromosomes.
Right.
Just as a shoelace without a tip becomes frayed and unusable, a chromosome without telomeres becomes unstable and can lead to cell death or malfunction. The Mad Scientist's Guide to Telomere Elongation. As a scientist in a lab, you're concocting a potion that can rebuild the worn-out tips of shoelaces' telomeres. This potion is akin to the enzyme telomerase, which adds material to the ends of telomeres, allowing cells to divide without aging. Your challenge is to create a potion that's strong enough to rebuild the tips, but not so potent that it causes the laces to grow uncontrollably, leading to a pile of tangled laces cancer. Nanoparticles, the protective armor. Now, let's switch gears to nanotechnology. Envision a knight's armor, but on a microscopic scale. These tiny suits of armor are nanoparticles designed to shield the knight, our cells, from the onslaught of arrows, radiation.
Right.
These nanoparticles can be engineered to reflect or absorb the arrows, preventing them from reaching the knight and causing damage.
Right.
The child's play of cellular mechanics. For a child's understanding, think of a video game where you have a character with a health bar. This health bar gets shorter every time the character gets hurt. In our bodies, telomeres are like this health bar, and as we get older, the bar gets shorter. Scientists are like game developers who are trying to create a special power-up, telomerase activation, that can make the health bar stay full longer. The Symphony of Nano-Acoustics. In the novel idea we discussed, imagine an orchestra where each instrument, nanorobot, can play a note, acoustic signal that reaches only a specific group of musicians, stem cells. These notes can tell the musicians to play louder, softer, or stop altogether. This is similar to using acoustic signals to control stem cells, turning them on or off for regenerative purposes. Bringing it all together. To apply these ideas in practice, we could develop a treatment regimen where patients receive nanorobots programmed with the right notes, genetic instructions to activate their musicians' stem cells at the right time. This could lead to targeted therapies for diseases, enhanced tissue regeneration, and perhaps a step closer to the dream of significantly extended lifespans. These metaphors and explanations aim to provide a multifaceted understanding of complex scientific concepts, making them more memorable and easier to grasp. If you'd like to delve into any of these analogies further or explore additional aspects, feel free to ask.
That's all for now. How much can I learn in one day? A lot. 20 minutes.